at the same university. He was a director and professor at Dr. Babasaheb Ambedkar Marathwada University, and currently he is working at the Jawaharlal Nehru University as professor. I welcome you, sir, to kindly deliver your talk.
Good afternoon to all. Our group works on antimicrobial and anti-cancers, especially from the medicinal plants and the beta-lactams inhibitors. So today I am going to speak one of the aspects that we deal, that green synthesis of silver nanoparticles, which are doped with the phytochemicals and their potentiality as an anti-cancer. So as we all know, this is about the size, and there are examples of biomolecules which are in the size range of the 1 nanometer to 100 nanometer that covers a variety of liposome dendriner, gold nanoshells, contour dot, fullerene, and that covers basically the glucose all the way up to the bacteriophages and certain viruses also. So there are various metal nanoparticles, or metals have been considered as a material for reduction into production of nanoparticles. However, the gold and silver have received quite a good position due to the fineness in the structural form and the dispersion of the nanoparticle per se. Otherwise, you can see like copper is there, iron is there, many of the divalent cations as well as trace elements have been considered. And they also have a different kind of physical properties as a metal and as a nanoparticle. There are different physical and the chemical properties. So the size spectrum, if you look into that, it starts with the 2 angstrom, that is a water molecule, and all the way the protozoans, which comes to the 2 micron, and hemoglobin to the viruses, that is where the biological range of the certain molecules. When we talk about the activity, bioavailability, it basically covers the bioaccessibility, absorption and transformation, multiplications of which. Now, bioavailability talks about the material to be made available, solubilized form, and its liberation to be able to make an effect to the site of action. So absorption basically allows the transport across the mucous layer and epithelial cells, and transformation basically to bring about the reaction so that the chemical and biochemical reactions would result into the favorable or unfavorable interactions. So there are two approaches, top-down and bottom-up, and as we know that the top-down would break the metal piece into the small molecules, finally into the exfoliation as a multi-layers or monolayers, or into the nanoparticle. On the other hand, bottom-up approach will start with the precursor, and all the way it will produce the condensed matter that is into the nanoparticle, and they could have a different type of structures, diamond, graphene, nanospheres, nanosheets, the nanowires, the fullerenes, etc. are made available. Number of nanometers we are very well aware of. So when we talk about the metallic synthesis, here especially the green synthesis part includes the biological material. Rather, you can have a physical as well as a chemical approach for the synthesis of nanoparticles. So plants and microbes have been so far tested, as well as recently the insects have also been used as a material for synthesizing the nanoparticle. And we could consider leaves, roots, flower, fruits, etc. are a part of the plant, or the whole microorganism, or bacteria, or fungus, or the algae extract per se as a material. That would be the blend of it as we use yeast extract in our laboratory media preparation. So this is an approach where you have to collect the plant material and make it into the small pieces, of course by surface sterilization, washing properly. And then heat as a principle. You can mix it with the particular silver or gold. Here we use the silver nitrate, and then you will get a change in the color which indicates there is a property change, and then you can characterize the matter in that changed color, and that's where you can know the nanoparticles are synthesized and what type of nanoparticle structurally and then to the functional part. So basically the extract contains all sort of secondary and primary metabolites. You can have proteins, you can have amino acids, you can have oligopeptides, you can have alkaloids, you can have flavonoids, you can have terpenoids, vitamins, sugars, and lipids also, and certain popular enzymes which then themselves work as oxidoreductase class of the enzymes. So that all material is available. So one has to figure it out what exactly is participating in the process of reduction and what is retained on the nanoparticle. Otherwise, synthesizing nanoparticle with physical, chemical or biological means at par same. However, use of grain synthesis approach is meant to reduce the energy and the thermodynamics required for A, and the second, the efficiency. Because often the natural compound that we know, a few of them have been known, just like the and there are at least 9-10 different chemotherapeutics used in cancer are actually the plant-based. But we still try to find more of the phytochemicals and more of the natural compounds because they will have a more bioabsorbability and bioavailability, therefore with a reduced side effects, because the drug used in the cancer therapeutic, one of the biggest downside is the side effects, and therefore the looking forward for the natural product is the business currently ongoing, and therefore there is a huge amount of side. And the nanoparticle particularly by themselves are the small, they are the delivery vehicle molecule. They will selectively collect the blend of phytochemical which is really and truly very small in size. So as of now what the molecules we know by different mechanisms, here also yet another way to know which all other small molecules or the very micro nano sized molecules we can figure it out. Those will bring an impact or make an impact onto the anti-cancer properties. So types of gold nanoparticle, as we know, gold has been very well said because the smallest nanoparticle, the finest nanoparticles so far known are from the gold up to 5 nanometer and mostly spherical, or if you can play around with the ratios and the temperature of the protocols, you can change the type of the size and the quality of the nanoparticle with the gold as well as with the silver. So the mechanism of synthesis, as I mentioned, that there is a blend of phytochemicals present in the extract, and that would interact with the metal ion, and then it would result into the production of, basically the reduction results into the production of the nanoparticle. So it has already been invoked to have several sort of applications, includes the various types of cancers, brain, skin, lung, breast cancer, prostate, colorectal, bladder, and photoimaging, cosmetics. There are a large number of cosmetics available from over 4 decades that contain the nanoparticle. Before even we know it, nanobiotech technology does have a big potential. We have been using lots of lotions that contains already the nanoparticles. So biomedical application, catalytic applications, food industries, everywhere you can see it has been used as a potential, and one of the aspects that I already told that we also work as a nanoparticle for antimicrobial potential. So when we talk about the prevention of bacteria, basically it either destroys the bacterium per se or targets one of the subcellular organelle, so that the bacterial replication or survival is jeopardized. Ultrasensitive amyloid detection is also been made as a sensor material. Then pathogen removal from the blood, that is septicemia particularly in that case, or you can say like parasitemia also, there also you can have an application. But here particularly I would be focusing more today about the advanced drug delivery, because nanomaterials and its application in terms of the natural products to deliver, because every metal ion if we use has its own localization, and thereby your phytochemicals will be easily traveled all the way with this nanoparticle to that site of action, and there you could be able to get its reaction as effectively as possible. For example, aluminiums are known to disrupt the cell wall. Bismuth is known to alter and modify the process of Krebs cycle, that is bioenergetics. Particularly in the case of gold, gold nanoparticle, gold is known to disrupt the cell wall as well as the DNA inhibit, it intercalates between the DNA like that dyes does, and DNA replication and transcription is prohibited. Silver nanoparticle, on the other hand, rather than intercalating, it affects the topology of the DNA. So rather than sitting into it, it basically affects the structural organization of the DNA and prevent therefore the DNA replication process. So in both the cases, gold nanoparticle or the silver nanoparticle, what is expected is replication is going to be prevented. And in addition to that, electron transport chain, as like in the case of bismuth, silver nanoparticle has an advantage because that's mostly important process in the biology where the ATP synthesis and ATP utilization is carried out. So energy transfer and the DNA basically duplication is going to be affected. So choice of silver makes justifiable. So one of I will be talking about two different plants. The first plant is Solanum xanthocarpon. As by the photo you could see that it is available everywhere, all parts of India, especially the dry area or the semi-arid area. It is quite rich with this plant. So when we synthesize a nanoparticle, we checked with the 10, we can see that around 22 or 20 nanometer was the average size, which again fits into the standard size so far documented for the silver nanoparticle. 10 is the smallest and up to 30 nanometers is considered to be the good quality silver nanoparticles. They were all spherical monodispersion that was seen. Then XRD was basically to support that the dispersion, and there is no monodispersion and there is no agglomerization or culmination of the different nanoparticles. Septine also shows that there are quite a few OSCH, then NHCF, carbon-carbon double bond, carbon-carbon single bond, like functional groups are available which have known impact on the biochemical range of reactions. Here the whole fruit extract, then whole fruit extract nano as a reducing agent in nanoparticles were synthesized. Then nanoparticles were biofunctionalized either with aspartate or with the arginine, and the chemically synthesized nanoparticle, that is basically the sodium citrate as a reducing agent and purified. So that's a very standard one which is known. So all the way up to 800 microgram per ml, the concentrations were used, but it is very clear that the first two, the whole, if you can look into the 600 and 800 nanometer particularly here, the whole fruit extract has a more inhibitory activity, and the second comes the nanoparticle. But when you have the nanoparticle coated with the aspartate or coated with the arginine, which is a very well documented biofunctionalizing amino acids in the literature, they miserably failed. So it's not a rule of thumb that something is published somewhere, it is written and it would always work the same way. So this is where we could confirm that the extract seems to be far better, even better than the nanoparticle. So it's not that every plant you take it and you will be synthesizing a nanoparticle and making it as anti-cancer. That's also not true. So this was a message wherein the nanoparticle can be produced only from the fruit extract, whereas we tried from the roots, we tried from the leaves, we tried from the stem. That part either did not have activity there itself, or nanoparticles were also having no activity. So this is only the part where we could see the activity. So the column chromatography was carried out initially too, because I told my student not just to document the nanoparticle based activity, just go further down and go more into detail and try to figure out what particularly the phytochemical it is there. Otherwise, what normally somebody will do is process the extract and know what sort of phytochemicals are present. That will give you qualitative or quantitative, but it will never tell you what is a reducing agent and what is doped on the nanoparticle. So that was something she had to do it. So column chromatography, 64 and 65th period did have the absorbance around 420 nanometer that resembles to the silver nanoparticle. So she processed the 64th number elevate further for the synthesis of nanoparticle with that, and then the activity was checked. Obviously that did retain activity, but again that elevate is not a fine purified compound. It may have more than one compounds or a blend of compounds. It is something better than the original crude extract. So she had to further analyze and there she had done it with the HPLC. The same eluate had to be processed then purified further with the HPLC. So there she got the 3 different bits at 3 different retention time points, and all those 3 she had to process to synthesize a nanoparticle and further use those nanoparticles for the biological activity, for the anti-cancer activity. So that was shown when she carried out the analysis. You could see that quite a few number of various compounds were doped in the crude extract of the whole fruit extract, ponisopridine, and the glycine, glycine, alanine, and glutamine, glycine, and alanine were the tripeptide. This was very unusual, like dipeptide and tripeptides, which have been considered presently as a short peptide as a therapeutic, as antimicrobial as well as anti-cancer. So that was also coming along with this, and then chlomipramine as well as ponisopridine have been documented to have a role to play in certain cancers. So that was interesting. Then she also prepared the another characterization of the particles produced from the HPLC fraction 31. There also she could get more of the di and the tripeptide blend of it. So here it shows that oligopeptides seems to have a more role to play as an anti-cancer. So round-shaped monodispersing nanoparticles were synthesized using the whole fruit extract that played a reducing role as well as a doping agents on the surface of the nanoparticle. Anti-cancer activity seen both in the whole fruit extract as well as the whole fruit extract mediated green synthesized nanoparticle. However, the activity was more with the extract. Biofunctionalization of green synthesized nanoparticle with arginine or aspartate cause a loss of anti-cancer activity. So probably the molecule, those dipeptide were masked by this, particularly converted into tripeptide or tetrapeptide. Those have lost the functionality of the earlier doped molecules. Then tripeptide as well as hexadecylamine were confirmed to be a phytochemicals docked onto the nanoparticle, especially from the HPLC purified fraction. The second plant is the Tridax cockney. Again, this is quite common in a semi-arid area, and especially in Jain, everywhere you will see this plant, everywhere. It is commonly called as a coat button or a Tridax, as a daisy also. And is a species of a flowering plant in the daisy family. It is the best known as a widespread weed and the best plant. I personally had interest in this plant because since my childhood, in Marathi it is called Dagdi's of Fool or Dagdi's of Alamis. It is, in Hindi they call it a Jakham Jodi. So it is having an application in healing the wounds. So that I experienced in my whole childhood. Every time whenever there was a cut or there is a wound, it was used very robustly. You just have to crush the leaves and like bandage, you cover it up, and after 3-4 days mostly it would be recovered.
Where it grows all over India?
All over India you will find it. All over India. I'm not so sure about Kashmir part, but semi-arid and dry area you will find it very easily. In Jain probably on the ground it's 90% available everywhere. Okay. So, I thought like probably on the skin part or wherever the epithelial cells are there, probably it has an effect. So it was also considered as a noxious because it spreads very fast, as well as therefore it is also considered as a pest. So commonly used in Indian traditional medicine as anticoagulant, hair tonic, antifungal, insect repellent, bronchial cataract, diarrhea, dysentery, and wound healing. So I only knew wound healing, but after reading literature from the Indian text, I could figure it out. It also has been implicated for a systemic application. So there is no harm in trying something else on beyond the wound part or the skin part. So nanoparticles were synthesized, and those were in the range of 20 to 25 nanometer in the size. Again, monodispersion, spherical, shaped, spherical to oval-shaped nanoparticles. XRD shows again there is no contamination or there is no kind of agglomeration of the nanoparticle. Zeta potential also shows that 20.7 millimoles, so that shows the stability of them. And we have been using the nanoparticle preparation for almost as old as a 3-year-old and having no loss in activity. So FTR here also shows carbon-carbon single bond, NH, CE, OH, etc. All functional groups are essentially present in the extracts. Then the MTP was carried out with the A549, even the previous also it was used on the A549 lung carcinoma cell line. And here it is clearly seen that when we have the nanoparticles, then the middle one is the extract, and this is a chemically synthesized nanoparticle, and this is the silver nanoparticle. So at 42.32 microgram, it was having the IC50 value. So that's a quite impressive as compared to the previous experience that we have had with other plants. And vinblastine has been used as a control model. So here we could see that silver nanoparticle has very high IC50, that is 11,596 microgram, while the TNP, that is Tridax nanoparticles, IC50 is impressively small, that is a 42.7, whereas the Tridax per se has a 40,672. The scenario is reversed than that of the Solanum xanthocarpum. Solanum xanthocarpum plant had activity in the fruit extract. Nanoparticle did have, but a reduced activity. Here, the nanoparticles have impressive activity. Plant is very bad as compared to the chemically synthesized. But even the chemically synthesized are 900 times weaker as compared to the green synthesized nanoparticle. Now, biofunctionalization was carried out again with the aspartate yellow color and the cyan color, the arginine, and it is consistent that biofunctionalization does not necessarily work even in these systems. So whatever phytochemicals are there, if they get in contact with this arginine or aspartate, they may produce ionic or covalent bond and changes the structure, thereby the functionality is no more retained. So here is IC50 again, 11,596 for silver nanoparticle, 41 is for the 42.7 for the nanoparticle TNPs, 40,000 plus for the Tridax, then aspartate 6,000 and arginine 1,000, but still it's far lower than that of the nanoparticle portion. So gradient silica gene chromatography was carried out for the further purification, as I told you in previous. The fraction 63 was processed further, and like in the previous case, the whole aqueous extract nanoparticle synthesized where HRMS was carried out to figure it out. So there were like 12 different compounds in the found onto the different type of nanoparticles, and ponisopril as well as tripeptides, already I have said that they have indicated to have a role to play in certain cancerous cases. So HPLC purified fraction was also utilized, and then aspartate, tyrosine dipeptide as well as the glutamic acid modified beta aspartate was also found. So interestingly, in both different cases we have been able to find di or tripeptide either as amino acid as such or the modified amino acid at end terminus. So clonogenic assay was carried out using different concentrations, 2 microgram, 5 microgram and 10 microgram. It is evident that at 10 microgram there is no growth at all, and the concentration dependent, the effect is noted just 2 or 3 minutes. Okay, trypanoside exclusion assay has also been carried out to know the cell viability at different time point, 24 hours, 48 hours. And here is also another cell line, that is a mouse skin melanoma cell line, that also it makes a very huge impact. And IC50 value is around 5 to 7, in between 5 to 7 micrometer, even more impressive than that of the lung particularly. Now we have gone further to figure it out at molecular level, what are the genes or what are the pathways or what are the processes being affected by the nanoparticle in the cancerous cells. So this is to show that the RNA sequencing was carried out, and there have been promisingly 32 genes, of which 14 genes are upregulated, while the 18 genes are severely downregulated. These are the highly stringent approach used to figure out the lesser number of genes. Otherwise, there are a couple of hundred genes which are upregulated and a couple of hundred genes which are downregulated. These are highly promising. And when we study the cake pathway, we can see that The KIP1 and KIP2 is also targeted, which has been known to have a role to play in anti-cancerous property. Then GP-SAR coupled receptors, that is also one of the targets by this nanoparticle. Then the third is collagen, so that is also the ECM matter is also being affected in the cancerous cell. Then also the metallothionein, particularly zinc transport, and zinc has been known to play a significant role in the transcription receptor, so that is also a part of it. Here are the processes. There are more than 30 different processes being targeted by the nanoparticle. Here are the financial assistance from the DST and SCRB to be acknowledged. The cancer work initially we started with the help of Professor Anna Pratas's group, and Rohini has completed her PhD. Now Nilesh is completing his PhD on the same topic, and Subhi is a postdoc working in the same area. Thank you for your patience.
So I think in the interest of time, I would request the audience to hold their questions for the coffee break. Thank you so much, sir. And again, I would request Professor Anju to kindly finish.